Hello everyone, we are from Group 4 and today we will be presenting our Scorpion Primus assignment. So these are our group members. For the first question, we need to do cDNA microarray hybridization to identify the genes involved in the production of taxol in Texas brevifolia. The steps involved in the experiment will be later explained in the next slide. The first step of this experiment is to collect sample, which is two samples of Texas brevifolia, one with treatment, which is metheljasmonate, and one without treatment. Next, we have to isolate the RNA through organic extraction method. After vortex and centrifugation, we will obtain aqueous phase, which has the RNA in it. Then we have to isolate mRNA from the RNA by using poly-T beads to bind with mRNA that has poly-A Then we can proceed to cDNA synthesis. Here we need to label nucleotides with two different types of fluorescent dye, which are red dye and green dye, to differentiate between samples with treatment and samples with no treatment. Then we can proceed to add mixture of poly-T primer and reverse transcriptase to proceed with cDNA synthesis.
Now let's continue with step 5, which is hybridization. The label cDNA applied on microarray slides. Then the cDNA molecules hybridize with the complementary single-stranded DNA. Wash microRNA slide to remove unbound cDNA. Then we go to scanning. Place the microarray slide using a microarray scanner. Result will show genes expression levels. Then after that we go to the data analysis. We use appropriate software to identify differentially expressed genes, and we compare expression level between Texas brevifolia metheljasmonate and Texas brevifolia no treatment.
So, in order to confirm whether the identified genes are truly involved or not, we can observe for the gene expression through fluorescent dye changes in the microarray analysis. We also can verify the upregulation or downregulation of the gene of interest upon the treatment. As shown in the figure below, we can see the red spot indicates the upregulation of the expression, while for the green spot, it indicates the downregulation of the expression. As for the yellow spot, it shows the constitutive expression for both genes. Moreover, we also can conduct qRT-PCR in order to confirm and validate the gene expression observed in the analysis. So that's all from me. The next presenter, please.
Quantitative real-time PCR must be done for the sample, which is Texas brevifolia. It needs to be treated with metheljasmonate, and control Texas brevifolia is isolated to be used as sample for qPCR reaction. After that, we need to prepare reaction mixture that contain forward and reverse primer, tag polymerase, magnesium chloride, the NTPs, PCR buffer, and also scorpion primer extract probe. And after that, we need to add the master mix into the samples containing mRNA template. Briefly centrifuge the tubes and put it into qPCR machine to run the samples. After that, the results are produced during each of qPCR cycle and the data will be shown in graph form. We also need to analyze and interpret the graph and the signal produced while running qPCR, which means the high peaks show strong signals were detected and indicate greater targeted amplicons produced at that time.
Okay, so for question 2, consider the detection method that is assigned to your group to quantify your expression and provide the advantages and disadvantages of the detection method. So in this case, I'll be saying about the advantages. Scorpion primers integrate the primers and probe into a single molecule. It provides excellent sensitivity and specificity. Scorpion primers can be multiplexed by utilizing different fluorescents, dyes, or probes in the same reaction.
You can see here, Scorpion primers do have a lot of advantages, but it also have the disadvantages, such as Scorpion primer design need carefully consideration of the stem loop structure as well as optimization of target specific sequence, which might be more difficult than building standard PCR primers. And also, because of the added component in the Scorpion primers can be more expensive than regular PCR primers.
Differences between quantitative real-time PCR and normal PCR is for qRT-PCR, provide quantitative data, detect by using fluorescent dye or probes, high sensitivity, less prone to false positive.
For normal PCR, it can provide qualitative data. Normal PCR products are most commonly analyzed by agarose gel electrophoresis. It also has low sensitivity and prone to false positive. That's all from our group. Thank you.